Hello students, in the previous video I explained about interkinesis. Now in this video let's start with meiosis-II. So meiosis-II is similar to mitosis, but there are some differences between mitosis and meiosis-II. Those differences I have given in the previous video, please go through it. So now let's start with meiosis-II. So meiosis-II involves two phases. They are karyokinesis-II and cytokinesis-II. Karyokinesis-II is divided into four phases again. They are Prophase-II, Metaphase-II, Anaphase-II and Telophase-II. Okay, so now the daughter cells of meiosis-I enters into meiosis-II. That is karyokinesis-II. That means they enter into the first phase of karyokinesis-II, that is Prophase-II. So these are the two daughter cells of meiosis-I which become the parent cells of meiosis-II. So now let's see what are the changes that occur inside these two cells during meiosis-II, that is during Prophase-II. So during Prophase-II, inside the nucleus, actually chromatin is present. Chromatin fibers, genetic material is in the form of chromatin fibers. Those chromatin fibers starts condensing into chromosomes. So they condense into chromosomes and each chromosome is with two chromatids. Hence this condition of chromosomes is called as dyad condition of the chromosomes. Okay. And nucleolus present inside the nucleus disappears and nuclear membrane also starts disintegrating by the end of Prophase-II. Now inside the cytoplasm of the cell during Prophase-II, all the cell organelles disappear like mitochondria, chloroplast, endoplasmic reticulum, Golgi bodies, all the cell organelles disappear. And the centrosomes which are duplicated during S phase, now they form asters inside the cytoplasm of the animal cell. But in case of plant cells, there is no formation of asters. This we already discussed. Again, I am going to explain about how these asters are formed inside the cytoplasm of the cell during Prophase-II. So, aster formation. So, one centrosome, this is one centrosome. One centrosome consists of two centrioles. Around these centrioles, around each centriole, macules are present. These macules work or function as MTOC, microtubular organizing centers, which form microtubules. And these microtubules which are formed are radiating around the centrosome. Now these radiating microtubules are called as astral rays. Now these astral rays along with centromere are called together form one aster. So inside the animal cell, now how many asters are formed? Two asters are formed per one animal cell. Now these asters, they start moving away from one another and by the end of Prophase-II, they reach towards two opposite poles of the cell which represents the spindle pole of the cell or spindle apparatus, from where spindle apparatus will be formed. So these are all the changes that occur inside the cell during Prophase-II. Now cell enters into Metaphase-II. Okay. So during Metaphase-II, there will be no differentiation between cytoplasm and nucleoplasm because by early Metaphase-II itself, the nuclear membrane undergoes complete disintegration. By the end of Prophase-II, it starts disintegrating nuclear membrane. By the early Metaphase-II, it undergoes complete disintegration and hence there will be no differentiation between cytoplasm and nucleoplasm of the cell. Right. Now, by the early Metaphase-II, formation of spindle apparatus is completed. By late Prophase-II, formation of spindle apparatus is initiated. By early Metaphase-II, formation of spindle apparatus is completed. Now, after completion of formation of spindle apparatus, the spindle fibers of the spindle apparatus attach with chromosomes at the point of kinetochores. After attachment, they bring these chromosomes towards the center of the cell, forming metaphasic plate. Now, this is called metaphasic plate. But in case of metaphase-I of meiosis-I, bimetaphasic plate is formed. Bimetaphasic plate is nothing but alignment of pairs of homologous chromosomes at the center of the cell is called as bimetaphasic plate. But here these are not the homologous chromosomes. Here there is no alignment of two chromosomes, homologous pairs of chromosomes, But here there is alignment of chromosomes, only one chromosome at the center of the cell, forming metaphasic plate. So we already discussed in previous videos while I was explaining about metaphase of mitosis, I explained about two different types of spindle fibers which are formed in spindle apparatus. Those two different fibers are, one type are continuous spindle fibers or pole to pole spindle fibers which run from one pole to the other pole and which are not attached with the chromosomes. These are called continuous spindle fibers or pole to pole spindle fibers. Other type of spindle fibers are discontinuous spindle fibers or chromosomal fibers or tractile fibers. So as they are attached with the chromosomes at the point of kinetochores, they are called chromosomal fibers. Okay, so after formation of metaphasic plate, now cell enters into Anaphase-II. So in this condition also, Metaphase-II condition also, each chromosome is in the form of dyad condition. Dyad condition means in each chromosome two chromatids are present or two DNA molecules are present. Right, now cell will be proceeded into Anaphase-II. So during Anaphase-II, at the beginning of Anaphase-II, centromere splits. After splitting of centromere, the chromatids of chromosomes start separating from one another and they move towards two opposite poles of the cell. This movement of chromatids towards two opposite poles of the cell is taking place with the help of interzonal fibers. These are interzonal fibers. With the help of interzonal fibers and even chromosomal fibers. So these are interzonal fibers, these are chromosomal fibers. We already discussed what are these interzonal fibers. I explained about these interzonal fibers in the anaphase of mitosis. Right. So movement of chromatids towards two opposite poles occurs with the help of interzonal fibers and chromosomal fibers. These interzonal fibers undergo expansion and push these chromatids towards two opposite poles of the cell, whereas chromosomal fibers undergo disintegration and pull these chromatids towards two opposite poles of the cell. So like this, these chromosomes are moving towards two opposite poles of the cell, and during this movement centromere is at the leading edge and arms are trailed behind the centromere. This and all we studied in anaphase of mitosis. Okay, by the end of Anaphase-II, these chromosomes having one chromatid are moving towards, they reach towards two opposite poles of the cell. So in this Anaphase-II, each chromosome is with only one chromatid. So this condition is called as monad condition of chromosomes. Monad condition of chromosomes. Okay, now after Anaphase-II, cell enters into Telophase-II. So what happens during Telophase-II? The chromosomes, by the end of Anaphase-II itself, they have reached towards two opposite poles of the cell, and now these chromosomes undergo decondensation into chromatin fibers. So like this, they undergo decondensation into chromatin fibers, then nucleolus reappears. Around this chromatin material, nuclear membrane is reformed and all other cell organelles are also reformed, and spindle fibers undergo disintegration. So this is about telophase. So by the end of Telophase-II, two nuclei are formed in each cell. After formation of two nuclei, then cell enters into Cytokinesis-II. So cytokinesis is nothing but cytoplasmic division. So this cytoplasmic division occurs by the formation of furrow in case of animal cells and by the formation of cell plate in case of plant cells. So in animal cells, furrow is formed towards the periphery of the cell. Furrow is formed in the cell membrane towards periphery at the equator. At the center of the cell, not here or here. So furrow is formed at the periphery, is formed in plasma membrane at the periphery and at equator. And this furrow deepens into the cell, dividing the cell into two daughter cells. Like this, cytokinesis occurs in case of animal cells. That is from periphery to center. As this furrow is extending from periphery to center, This type of cytokinesis is called as centripetal cytokinesis. We already discussed in the previous video. And in case of plant cell plate is formed at the center, and that cell plate will be extending towards the periphery of the cell, dividing the cell into two daughter cells. So as it is extending from center to periphery, cell plate, it is called a centrifugal type of cytokinesis. So by the end of cytokinesis, two daughter cells are formed from each cell. That means totally 4 daughter cells are formed. These 4 daughter cells are haploid. These 4 daughter cells are haploid, and the number of chromosomes and the amount of DNA present in these daughter cells, each daughter cell is half the number of chromosomes and half the amount of DNA present in its parent cell, which enters into G1 phase of meiosis. G1 phase of the interphase. That means which enters into meiotic cell cycle. So this is all about Meiosis-II. Meiosis-II. In the next class, I will be explaining about the significance of meiosis. Thank you, students.